thanks to Pfenex, and in particular Cassidy and Christy and Bill for assembling the discussion and the invitation. So just as Bill said, Matrivax, we are a very local company. We're in Boston. We're located in incubator space nearby in the Boston University area. And I'll tell you about our story about our virtual conjugation technology. So just one slide about kind of the fiscal makeup, if you will, and the technology, the background. Company overview format. We have three founders and board of director members. First and foremost, John Mechalanos. This is really his technology from Harvard Medical School. John's a luminary. He's made live attenuated a vaccine headway and turned his attention towards subunit vaccines, and what I'll tell you about the technology called Protein Capsular Matrix Vaccine. Yi-Chen Liu at the School of Public Health and a connection to VTI, Vaccine Technology Inc., which is sort of like a very similar company to ours. We're both under the umbrella of Gerald Chan, who's at Morningside and very interested in vaccines both in China and in the United States, a leading investor in the vaccine industry. Our company, as I said, we are located in Boston. We got on the ground, I should say formed in 2007, but on the ground in March of 2008. We're now up to 12 people. And I think the technology we have falls into this general heading of virtual conjugation. What does that mean? When you think of a conjugation, it literally is the first one commercialized, Haemophilus influenza B, the Hib vaccine. You have a polysaccharide and you molecularly couple it to a protein, and there really is a direct physical link. But the key molecular event you're trying to do is invoke T-cell help. It doesn't necessarily have to be physically coupled. Virtual conjugations are those that are conjugations or polysaccharide protein in proximity, but not necessarily molecularly linked. It's a bit of a nuance, but I'll kind of show you a schematic later that I think will make it quite clear. And we do have this strategic alliance, as I mentioned, with VTI, this company. They've been very helpful with us as we move towards process development in GMP to when things can get very expensive. When you move to a CMO, we have sort of worked out the bugs, so to speak, with VTI at their Haikou facility in China. So we are living in what's called the Gilded Era of vaccinology. And I think this slide and these data very much support that. If you look at global vaccine market growth, step out here, in particular, this period here, when compound annual growth rates were about 10%, in the chemical industry, in the new chemical entity industry, the rate was not nearly as high as 10%. This is really profound growth. You look at vaccines in billions, we're now above the $20 billion mark. Why is that? Certainly during that high slope blockbuster vaccines, that was when Merck introduced, for example, Gardasil, the papilloma vaccine, clearly is part of the area under those curves. We have vaccines that I was involved with at Rotarix, at my former company, that got licensed to GSK, Rotatech, so rotavirus vaccines that had a big play there as well. But there's other factors too. Price cap removals, that's really helped the flu vaccine industry to make it less of a commodity market. Growing the middle income markets, very important as the BRIC nations of Brazil, Russia, India, and China begin to emerge as an emerging middle class and people who can now afford a vaccine and pay a going rate for it. And the developing world demand, where the Gates Foundation has come in and really put in a mechanism of how companies can actually make some profit, not a ton, but some, for commercializing vaccines, or I should say manufacturing them before they're sold at discounted rates. The technology that I'm going to talk about, PCMV, is, I won't say specifically applicable, but largely applicable to polysaccharide vaccines. So when you look at this currently greater than $20 billion vaccine market, about a quarter to as much as a third of it are polysaccharide vaccines. And I left out one big one, or really two big ones here. In 2000 Prevnar 7 was introduced. Now we have Prevnar 13 as the best vaccine for efficacy against pneumococcus. And of course, there's meningococcal vaccine. So they are certainly very big players in these values. So now the transition to the polysaccharides or the polysaccharide vaccines. Number one, first and foremost, the biggest vaccine currently are those that are in the pneumococcal field worldwide. Despite having efficacious vaccines, excuse me, that were unconjugated, now conjugated since the year 2000, there's still 1 million deaths per year in the world. In the US, multiple 3,000 cases of meningitis, 50,000 cases of bacteremia, tenfold that of pneumonia, and hepatitis B as well. And this evolution is really what happened. Pneumovax is a vaccine that really has terrific breadth. It's 23 valent, but they're just 23 sugars that are mixed together. Highly efficacious if you're greater than 2 and under about 50. But if you really look at who's dying here of invasive pneumococcal disease, it's infants and kids less than 2 and elderly people above 50 years old. So the vaccine really didn't cover the true target population. So thus, there was a move towards vaccines, for example, Prevnar 13, which is honed in on the specific serotypes that, for example, are in US and Europe and a little bit of the world, and efficacious in those flanks, if you will, the adult population, excuse me, the elderly population and the infant population. And again, to that point of how valuable these vaccines are, just last year the sales were approaching $4 billion. And as Bill alluded to in the introduction, one of the big difficulties of this vaccine is making it. I mean, it's incredibly efficacious. It's clearly the most complex vaccine ever made. You have to have serotypes and all the different polysaccharides made in bulk, held in bulk, conjugated in bulk, moved together, and cocktailed together. So this vaccine, as manufacturing increases, while the market matures, is predicted to be upwards to $8 billion, with $5 billion in low to middle income markets that I talked about earlier, for example, in those BRIC nations. Second, again, these diseases are horrible, these bacteremias from meningitis, whether it's meningococcal or pneumococcal, and sure, very much the same evolution. There were unconjugated polysaccharide vaccines, in this case a quadrivalent vaccine that got converted into a conjugate vaccine. Matrivax now sells over $1 billion in vaccine. And Novartis recently launched Menveo, which is also a quadrivalent vaccine. And I show this third category of enteric fever vaccines, not because it's a big market, but because it's relevant to the talk that I'll talk about later, which is our proof of concept moving into the clinic. We're going with enteric fever. Nonetheless, there are currently efficacious vaccines against typhoid. Same story. Unconjugated polysaccharide works well in kids, or I should say people that are 2 years of age or older, but not in the target population most needed. And then an emerging disease, particularly in Southeast Asia, Salmonella paratyphi A, which is also encapsulated vaccine, but again quite different. And there's many deaths from paratyphi A, despite these vaccines that are available. So that's, like I said, the reason I mentioned typhoid is because it's one of the targets that we're going after first clinically. Okay, the technology. This slide, I really want to kind of point out that nuance that I touched upon in the introduction about how does this differ from a conjugate vaccine. So in a conjugation, the analogy that I think works real well is it's like catching fish with a hook. And as I show this kinetic diagram, you have these blue proteins, represented molecules, hooking directly to the polysaccharide. So this stuck to this. So really the key thing is you have intermolecular bonds between protein and polysaccharide. With a PCMV, on the other hand, you can see how this comes together. It's like catching fish in a net. So the intermolecular bond between protein and polysaccharide is an intermolecular bond between protein and protein. So it's like catching fish in a net so you're bringing the polysaccharides in proximity to one another, but they're not necessarily covalently coupled. That's the distinction. So that's shown for a monovalent vaccine. This could be the typhoid vaccine that we're making. But you really see the benefit, the applicability of the technology as you become more multivalent. And this, for example, could be a schematic of the quadrivalent meningococcal vaccine that I just mentioned to you. So in a quadrivalent meningococcal vaccine, the polysaccharides are first sheared to smaller size polysaccharides, and I can tell you that those are less immunogenic than large polysaccharides. We have some data on that. And there's a reaction chemistry for one linkage, covalent coupling, a second, a third, and a fourth. So these are all done individually, and then they're simply mixed together to make a conjugate vaccine. That's commercialized. So what we can do is take these same colored polysaccharides. We do not share them. We make in fact, for example, VI polysaccharide is in the megadalton range. These polysaccharides are typically $10,000. So our polysaccharides are anywhere from 10 to 100-fold or more larger. And we can take all of them, as represented by the different colors, and again, entrap them simultaneously. So here you have a single reaction and one chemistry, and here you have 4 different reactions in a few different chemistries. So you can see that the technology is becoming more simplified as represented in that schematic. How does this translate? Well, it translates mostly in one of the main axioms that we have for the company is it translates in cost of goods, clearly. We are not trying to make a better conjugate vaccine, because I have to tell you, Prevnar 7 in the US was more than 90% protective. So if the serotypes are covered, you get great efficacy. But what we are trying to do is to make, at least for those serotypes, make it a simpler process, build a simpler mousetrap. But where we can become more effective is by including more serotypes, and that's certainly our goal. So this is an example of what would be Prevnar 13, a 13-valent conjugate on the side. And these are estimates on there. But basically, by the time you do the 4 different chemistries of the 13 different polysaccharide linkages to a unique carrier protein, CRM197, there's about 90 or so steps to that. There are 4 different chemistries, you formulate. It's about 92 GMP steps. And this is really, you have to take this with several grains of salt. Because these are estimates on my part. But with the PCMV process, we could theoretically take 13 polysaccharides and lump them together. That's possible. I can tell you we're not there yet, and I'll show you data for that. But what we do see is potentially making bundles of 4, 5, or 6 polysaccharides at once, and that we cocktail together towards an 18-valent, 24-valent, what have you vaccine. And in this particular case, we see these number of GMP steps reduced to 5 to 10. Again, and our plan is to increase valency, and therefore, by that definition, have better breadth of protection, and also with a simpler process. There's something happened here.
Just click on the slide and then
Okay, so in kind of a quick review here, this is the evolution and where we are commercially with pneumococcal vaccines. Again, the 23-valent vaccine is known as Pneumovax. It's unconjugated. It's got the breadth you want, but it doesn't have the targeting for the person, right? It doesn't work in 2-year-olds or less, or 50-year-olds, 55-year-olds or older. First came Prevnar 7. Those are the 7 serotypes with those X's that are included. Next came Synflorix of GSK, which is a 10-valent vaccine. The difference is shown in red. And then the state-of-the-art of the vaccine now is Prevnar 13, which is the 3 added serotypes that are in yellow. And in fact, Merck, as we speak, has a 15-valent conjugate vaccine that is in clinical development, I believe, in phase 2. And then in the table to the right there, what I've done is done some kind of good guesswork based on looking at serotype prevalence in the world and in China. And the prediction is that a Prevnar 7 would give about mid-60% protective efficacy in China and the world. Synflorix, 80% in the world, 65% in China, and the Prevnar 13 in and around the 80%. So obviously far improved, but certainly a way to go. But if we had about a 24-valent vaccine, we could essentially get up to greater than 95, 98% protective efficacy. And if the PCMV technology works as the way we hope it does, we're going to try to get there.
Can I just ask a maybe very ignorant question? But so my understanding is that the lack of efficacy in those target populations, the under 2 and the elderly, is not a function of the number of
Well, in the world it is, right? But you're right, because it's not, in the United States, for example, Prevnar 7 is about 90+% efficacious. So clearly those 7 serotypes were highly selected and thought out about in the United States. So if you look at which serotypes are prevalent in the US, those are what you get. So you're right, there's great protection against the serotypes that are included. But obviously if they're not included, you're not going to get protection against them.
No, I guess I'm asking a different question. So I thought that where you're going here is by saying the more serotypes that you include, the more valencies you have, the wider your protection in the target population.
Yeah, well, so if you had a 23-valent vaccine that was conjugate or PCMV, you'd get probably 99% protection in the United States, right?
So I thought that the lack of efficacy in, for example, the elderly had to do more with immunosenescence and the suboptimal response versus not having the correct serotypes.
That's absolutely true. In the unconjugated vaccine, like Pneumovax, there's not very good data at all in the elderly. So the serotypes are there, right? They're all represented, but because you're not getting T-cell help, you're not really getting much protection. Pfizer has just now done a study with Prevnar 13 looking at what's called CAP, community-acquired pneumonia in the elderly, and shown that they're getting terrific efficacy looking at those 13 serotypes of pneumonia. So it all depends what state the polysaccharide is in, right? There's sort of two different issues. One is, does the polysaccharide work? If it can. The other question is, are those serotypes represented in the population that it's in?
That could be the question, the role that the carrier protein plays in driving that type of improved immune response in the very young and very old.
Exactly. Maybe not the carrier protein. I mean, CRIM is certainly a great one, but tetanus toxoid, whatever has, I'd say any protein that has multiple T helper epitopes would serve just well.
The carrier protein is necessary to achieve those types.
By invoking T help, yes. Okay, so we're a 12-person company, and although the most applicable target we could aim at would be pneumonia, we physically can't do it. So we have to just keep our pneumonia target on the preclinical horizon for the time being. But to show proof of concept and move towards the clinic, we wanted to pick something that as a biotech company we could get our arms and our head around. So we picked typhoid fever because it's a monovalent VI, which is the virulence polysaccharide from Salmonella typhus. And now this part of more the data part of the presentation. So these data are now a couple years old. It was our first foray into where would we stack up against an existing vaccine. So we did mice preclinical immunogenicity studies. We purchased Prevnar. In this particular case, it was Prevnar 7, because Prevnar 13 wasn't available. And we had a sense that once these particles formed, if we could size them, does size make a difference? And what we did here, for example, in pool 1, pool 2, these are different size fractions, and I can tell you about that pool 1 is about a couple hundred nanometer size particles. Pool 2, about 100. Pool 4 was the size of about a virus-like particle, 40, 50 nanometers in size. And when we did a dosing regimen, 3 biweekly doses, 0, 14, and 28, and looked at the immune responses, we were encouraged by what we saw. When we compared against Prevnar, which is here, now in Prevnar, this is looking at serotype 14 only. There's 2 micrograms of that amount constituent of serotype antigen in the vaccine. It elicited a titer of about 750,000. But if you look at our pool 1, which happened to have 0.03, which is less than 2% the amount of antigen, okay, so at about 1/50th the dose, we elicited titers that the GMT, these are IgG antibodies, that were 500,000. We thought that was pretty good. Titers were comparable, 500,000 versus 3/4 at 1/50th the dose. So, we were pleased with that. We thought we had some traction. And we then asked the question, well, this is more of a quantitative aspect, and ELISAs can be good, but they can be a little bit misleading. Are the titers that we induced, can they elicit an anamnestic response? and also did they have functionality. So the first question we asked was, do they elicit an anamnestic response or a memory response, which is critical. That's the type of response that you need in a young child, an infant. So we took those same lots of animals, and we waited for them. We waited for the titers to begin to decline. You can see that this was boosted something comparable. The titers went from 500,000. They finally started declining on day 239, which is about 8 months. They started dropping. And what we did is we boosted on that day compared to the polysaccharide by itself and looked either 4 days out or 3 weeks. And we're looking for IgG. And I can tell you that if there's no memory, we're going to get a lot of IgM. And if there is a memory response, at 4 days, you'd only see it with looking at IgG if it were in existence. And sure enough, you see the titers begin to rise 4 days out from 152 to 266, and then 3 weeks later, they get to about 860,000. Certainly very positive or strong correlation of an anamnestic or memory response. The controls behave just as they should. Polysaccharide by themselves, you can see, were flat, either after 4 days or 3 weeks out. So now we had a good sense that the immune response was present and that T cells indeed were invoked and that we got an anamnestic response. Without a T cell helper cell, you would not see an anamnestic response. Now looking a little bit differently at the functionality of the antibodies. Okay, they were memorable. They were high titer. Do they have functionality? So we worked on a challenge model in mice. It's tough to get pneumo to work real well. So basically we took the sera from those mice that were immunized, passively transferred to naive mice, and challenged them with about 1.5 times 10 to the 150 million organisms intranasally, and asked the questions versus mice that did not get sera transferred. Did they survive or did they live? Did they survive or did they die? The PCMV animals in the triangles, 100% protection. Looking out after 60 hours, I can tell you that they lived for a happy life. And by about 2 days out, 6 of the 8 mice that were in the control sera died. So this proves that the antibodies that were elicited were functional and conferred protection against challenge. This is kind of a roundup of those data. You've seen the latter 2 bullets. I can also tell you that a key in vitro measure to look at functional antibodies is what's known as the OPA assay, the opsonophagocytic assay. And sure enough, the titers that we saw in the ELISAs induced by PCMV were comparable to that induced by Prevnar 7. Okay, so that we felt comfortable about that. So about a year and a half ago now, at the company, Matrivax, we had really kind of a breakthrough, that's something that we learned, and it was the following. If you think about a polysaccharide, it's typically polyanionic. It's not terribly unlike DNA, particularly VI. VI polysaccharide is particularly polyanionic, negatively charged. So if you think about trying to use that analogy of catching fish in a net, if it's strongly anionic, even if you have CRIM around and you cross-link it, if it's not in and around where the polysaccharides are, you're just going to form a net that'll be empty. So we were getting low entrapment efficiency that we wanted to improve. So the idea was if we could negate the charge with a cation, could we cajole polysaccharide towards the to-be-made CRIM 197 matrix? That's the thinking. And we tried that, and what was good about it is it worked and it extends our patent life out to a substantial amount. We found that it indeed facilitated PCMV formation. Now we got an increased efficiency of polysaccharide entrapment of PCMV, which we theorized and hoped. But the last bullet there, which I'll show you data for, was completely surprising to us. It actually, in its true sense, worked as an adjuvant. It increased the immunopotency of a polysaccharide PCMV, which we were very surprised to see. So here's some data that it's nice to have a 30, 45-minute talk because I can actually show this because I usually have to exclude it. So in the studies that we had done the first 2 years of the company, they were more or less what you're looking at that top graph there. And what you can see, this is using VI polysaccharide as an example. We call these unoptimized captures. So basically the PCMV is in the dark blue, and the VI polysaccharide, again it's unfractionated, is in the light blue. And we were getting low entrapment, about 5%. And what was happening was, this is without any polycation. What was happening was, because of the high degree of not contamination, but free polysaccharide, only about 4% of what we're immunizing with is really what we wanted. It's entrapped PCMV. The other of it was just free polysaccharide. So we did two things. We extended the column length, no big surprise there, but you really see the difference here. So when we start adding one of the polycations, what we call MRD 517, suddenly the entrapment went from 5% up to 53%. Now what we were immunizing with was 72% VI that was entrapped. And then when we basically use longer columns, you can see we enriched for not a nearly homogeneous, but a greater than 99% set of particles. So now we're immunizing with the material that we really want to do, which is a VI that's completely entrapped in the polysaccharide and separated out from free polysaccharide. And this was really an issue with VI because it's so big. A lot of the other pneumococcal polysaccharides are not. So now we're beginning to ask the question, can we build towards making a multivalent pneumococcal vaccine? And we started out with groups of 3, just because we thought that was a reasonable goal. And here we picked 3 serotypes to put together, 4, 18C, and 23F. Again, a typical dosing regimen. By this time, Prevnar 13 was available, so we went head-to-head, an exact dosing regimen. We had comparable amounts of antigen, 2 micrograms and about 2, slightly less. And what we saw, we thought was pretty impressive. So this is with the MRD 517. Looking at serotype 4, the titers induced were more than an order of magnitude greater than Prevnar 13, more than 10-fold more, which is, here's a vaccine that's tremendously efficacious against serotypes. Looking at 18C, we had a titer of 76,000 versus Prevnar was 35, so well more than an order of magnitude, approaching 1.5 logs or more. And then 23F, we were about 5-fold lower. So we were very encouraged by these data that 2 of the 3 serotypes that we looked at in the PCMV were substantially higher than the Prevnar-induced titers, and 1 was less. So the board said, well, they wanted me to do this next experiment that I was holding my breath on. What would happen, so let me click on the slide again here. What would happen if we tried a PCMV with all of the 13 antigens that are in Prevnar 13 in one reaction, and could we get good immune responses? And this was obviously a very difficult challenge, but we tried. And what we got was actually not bad for the first time out. And so in this particular case, there's 2.2 micrograms, as I said before, each polysaccharide, except for 6B, in all of Prevnar 13. We had about 0.3 micrograms. So we have literally, about 10% or so, 15% the amount of antigen, and that's a guess. We did not have the analytics in place to measure each constituent polysaccharide. So this is just assuming that an equal amount got in, which of course we know is not true. So again, you have to take this with a grain of salt. Suffice it to say, at much lower antigen levels, if you look, for example, in the titers that are in red, for 3 of the serotypes we're doing better than Prevnar 13. In the black, we're kind of in the comparable level, and those in the blue are actually significantly lower or inferior to those immune responses. These data are an aggregate that kind of explain the multiple numbers of either bundled antigens that we put together, and this is where we are. So when we made TRI, and I showed you one piece of data, or quadrivalent bundles, and looked at, we were superior in immunogenicity for 4 of the antigens that were shown, for serotype 4, 5, 14, and 18C, and in the 13 and 23 valent 3, and so on. So collectively, if you look at all the data together, of the 10 antigens that we've looked at, or serotypes, we had improved immunogenicity for serotype 1, 4, 5, 14, 18C, and 19F. That's pretty much where the program stands now, but now we're trying to actually sort out which polysaccharides bundle best together. And it really is kind of remarkable. Sometimes if you put 3 or 4 together, one polysaccharide will include the other. Sometimes you'll add a different polysaccharide, it will almost act as a chaperone to bring them in. So there really is work to be done here to figure out the best bundling antigens that work together. Okay, so as I said, that's the preclinical side of pneumo as an example. That we wanted to take typhoid into the clinic, but obviously we had to start preclinically. And we thought this was a really good target because of the following. The existing vaccines from Sanofi and Typhoid are VI polysaccharide. And same scenario what I mentioned to you before. They work okay, 70% efficacy, greater than 2-year-olds. I honestly don't know what the efficacy value is in the elderly, but we'll call it, 18 to 50-year-old people. So the efficacy is okay, but it's not that great. No memory, not surprising, no anamnestic response. And there is a live attenuated vaccine called Viviteeth, which was Burn Biotech and now Crucell. That's is sold as either 3 or 4 dose capsules. And again, comparable efficacy to the unconjugated polysaccharide vaccines. So we worked out a lot of dosing since, but I can tell you we had early success with this. When we made a VI CREM197 PCMV using one of the polycations, which we code as MRD517. When you look at the polysaccharide by itself, which is comparable, similar to what Typhim or Typhoid would be, we get its reciprocal titer of 1600. When you make a PCMV out of it, the titer goes up more than 40-fold. So we're going to have a 40-fold increase. We felt good enough that was, good to take forward towards a clinical candidate. I mentioned in the opening slide that we have this alliance with VTI and the company in China. And originally our plan was to do preclinical stuff and really out the tech transfer in the US and then do everything in China thereafter. But the Chinese regulatory system is getting kind of worked out, and we really changed the pathway. So what we do is research all the way to tech transfer. And we do our scale-up, which has been very useful for us for making some CREM197 lots, mostly VI. So, because before we go to a US CMO, we have to start paying, sort of expensive dollars, if you will, to really work towards a clinical trial, it's good to have some of these things worked out at scale, which obviously we can't do. We're about a 10-liter level in Boston, and we can get up to several hundred liters, several hundred liters. At VTI Haikou. So that's been an important alliance for us. And then we go back to the US and make our GMP product here. So that's the pathway that we're actually following. Here's the pilot facility that we built together in Haikou. This structure was existing, and we went in really a year and a half time. That's a 3,000 square meter facility. It's capable of doing 3 parallel suites, 1 for a carrier protein, 2 for polysaccharides. It's got 50 and 150 liter fermentation capacity, and it was actually completed a couple years ago now and was fully validated. So we don't use it in, like I said, a GMP capacity. We use it mostly for, as I mentioned, scale. Okay, so here we are with our VI PCMV development at a GMP level. As I mentioned, that kind of pathway I just showed to you, that's exactly what we followed. Work out the details of Boston, scale up in China. It's very costly and very expensive to figure out in terms of the human resources to put towards these projects. This is what our technology is. It's making a PCMV, and we were getting a lot of attention having to go here. So after the lesson learned of using or making VI and figuring out how to reinvent the wheel of how to grow Salmonella typhi, express, purify, all the release assays, we came upon Pfenex, the host of the meeting here, and were able to come to terms really very quickly with, again, knowing what it took to get here to actually do a deal where we could just simply license the technology and CREM197 to go into our clinical trials, and that's exactly what we've done. So all this comes in a little bow from India, thanks to and there, like I said, there's a third component here. MRD517. We had a company in Canada, GMP produced that for us. Very good in polymer chemistry. And so this is released, this was manufactured at the GMP level just a few months ago. and we're scheduled in October to actually do the PCMV at a GMP level. We feel comfortable about this because we did do one trial run. We did a non-GMP run this past January, February, using VI, Crim 197, Pfenex Crim, and our material from Canada. And we were able to make the particle sizes just as we wanted at scale. And we did a sterility testing and endotoxin testing. All the release assays that we had in place were actually met. And this was the plan for the clinical trial, I should say is the plan for the clinical trial. We plan to go into 18 to 40-year-olds sometime in the middle of next year. Group sizes of 15, 2, 5, and 10 micrograms will include Typhoid VI. And what's really useful here is the data that we get, our phase 1 through 3, we'll obviously get safety data in a dose escalation format of the study. But also, it's well known that if you elicit, as Typhim and Typhorix do, 1 microgram per mil of IgG antibody against VI, that's correlated with protection. So we can get correlative protection type data here. That's why I say phase 3-like. So we're excited about this. And we did take our package of the non-GMP material, and we had our pre-IND meeting with FDA, which is a company first this past April, and ran the synthesis and manufacture by the FDA, showed them our toxicology study design, our plan to do in vitro potency as opposed to in vivo potency, and the clinical design I just showed to you, and release of stability studies. And by and large, they were satisfied with what we had. Of course, they'll have to wait to see the IND before the comments come in, but we were okay with what they said. In summary, so we inherited this technology called Protein Capsular Matrix Vaccine. It was good, it had good proof of concept looking at some of the pneumococcal and typhoid antigens. We had a very important discovery at the company a couple years ago and showed that the addition of polycations prior to the formation of the particles, making of particles, was instrumental both for polysaccharide antigens, pneumococcal antigens as well as VI, 40-fold higher. And the GMP manufacturer is now complete of our 3 components. As I just mentioned to you, we're scheduled to do the PCMV synthesis. I hope it goes as well as the non-GMP one did at the beginning of the year. And we look to start a trial in the first half of next year. On the pneumococcal side, we had some funding from the Gates Foundation to do work. We've tried all different size packages or bundles, if you will, with different antigens and putting them together. And 3, 4, 13, and 23 valent we started. And when we looked at it, we were quite excited by this. 6 of the 10 polysaccharides that we looked at immunogenically were superior to Prevnar 13. So we hope that this will lead to eventually a simplified manufacturing process so we can get vaccines at lower cost of goods and include more serotypes and have a greater breadth of protection. And with that, I'll close and take any questions you may have. Yeah?
For those serotypes or polysaccharides for which you aren't getting a good immunological response.
So you said, sorry, you said are or not?
Are not.
Yes.
Have you considered that it is because they're in the presence of other serotypes and that if you prepare them as singular particles or PCMVs, that in fact you could elicit a better, more in line with the way Prevnar produces them?
Yeah, so it's like almost like an antigen interference.
Exactly.
Yeah, in some cases we actually see that if you bundle the wrong bad apple in the bundle, if you will, you can actually almost see an antigen interference. Yet we haven't. It's really become quite empirical for us because sometimes that same antigen which was interfering against the immune response we thought of a couple that it was bundled with might serve as almost a chaperone to enable better incorporation into a PCMV. So at its face value, it can look like antigen interference, but we're not quite sure yet what it is. Yes.
What was the stability of the PCMV vaccine in the plasma?
Yeah. So that's a question that we will certainly we're dealing with as we speak. Because, in a conjugate, if you think about it, once you make that molecular hook of protein to polysaccharide, it basically ain't coming apart, right? It's a covalent linkage. And we're saying that we have non-covalent linkages. So we've taken, for example, I'll answer your question two different ways. We've taken, for example, VI polysaccharide, which has no side groups that could form a covalent linkage with CRIM-197 using the chemistry that we use. We've tried to electrophoretically separate it because these polysaccharides are so long, they must stick out like spaghetti strands. It's very, very difficult for us, very difficult to actually pull them apart. We can treat them with protease and break them apart. That's what, and release the sugars. So the assay that we have in place to look at that is a capsulizer. And what we've done so far is we've taken, the way the capsulizer works is we have antibody, a monoclonal antibody against CRIM that's bound to the well of an ELISA plate. And then we put on particles. And then we come in with a detecting antibody against VI, okay? And it's very quantitative, actually. And we've shown that we can keep samples at 4 degrees for a year and a half, and we see no titration of signal. Yet if we specifically try to break apart the particles, we'll see the signals goes down. So we can do the right controls and see as the particles, make the particles come apart and see a decline in signal. But at least a year and a half out at 4 degrees, they look really quite stable. And we intend to store them in liquid formulation. Yeah.
So what is the source for the polysaccharide? Is what is this?
What is the source?
Yeah, for the polysaccharide.
So for the VI, we make it ourselves. It comes right from the animal, so to speak, from Salmonella typhi. It's shed. It has a bit of a Poisson distribution. It's probably the average molecular weight of VI is a couple megadaltons, so it's in the million molecular weight. And it's very hard. VI is extraordinarily stable. It's hard for us to break it down. We're now doing some work with pneumo, and we're finding with some homogenizers, we can actually break pneumopolysaccharides down to smaller fractions. Yeah.
So have you considered making smaller pieces of those polysaccharides that are excluded so that if they have some difficulty, that may be reduced by smaller size and they will be entrapped better by the ones that you have no problem incorporating in the particle?
It's actually quite a good idea. Never thought of it, but it's certainly a fine idea. No, we have not tried that. Our thinking is, and it gets to the earlier question about stability, if we make a matrix of protein, at some point if the polysaccharides are too small, they would have to titrate and diffuse out over time. I mean, it sort of goes without saying. So that's kind of the next experimental parameter we want to start asking is, what size can we go down to where we actually get inclusion and entrapment? And your idea is a perfectly good one. Will that make a difference? I don't know. But then we have to also be careful on the flip side that because it's in there and entrapped, can it diffuse out over time? But your point is well taken. Yeah.
What do you estimate the unit cost of your vaccine would be for typhoid? Any ballpark figure for this?
No, I'll tell you, no. I'm very hesitant. I'm not being coy here. I'm very hesitant to give cost of goods because, we're at a 50-liter fermentation scale. We're a biotech company, and as I mentioned to the folks who do the work, is, we're not trying to make the most cost-effective typhoid fever vaccine. Our goal is to get enough material that we can put into a PCMV, do a phase 1, phase 2 clinical trial, and that'll be good. We can go back and sort of optimize here and there. For example, as we scaled, like those data that I showed to you in 2 mil reactions, we were getting 50, 60% entrapment efficiency, which is outstanding. Some of the conjugate vaccines at scale, they may only get 5% conjugation. That's with a covalent process. But sure enough, as we went up to 50, 100 mil, much greater scale, now we're down at the 10% level of entrapment. And I think there are things we could do to get that back up, but we really haven't pushed very hard in that domain yet. That's why I'm I'm really hesitant to even make that calculation now. Yeah?
I'm sure everyone in the room here has had vaccines made from killed, attenuated, or live viruses or bacteria.
Yeah.
So when we talk about 13-valent or 24-valent, or why has it gone that way for a lot of these products instead of just doing it the way they used to with whole viruses and bacteria?
That's a great question. And I just saw some data at a meeting about a year ago from Rick Malley. He's local at Children's Hospital, and he asked that question: why not make a whole cell vaccine against pneumo? Well, it has pneumolysine. There's lots of toxins in strep pneumonia, so he was asking some proof of concept questions. And not really removing any of the toxins. And sure enough, once they get up to doses that were immunogenic, they were pretty reactogenic, I can tell you. So, although it's a gram, let's use strep as an example, it's a gram positive, there's no endotoxin per se, but there's still teichoics and acids, and, capsule really isn't, it's a really poor immunogen. So you obviously want to get the capsule responses elevated. To do that, you have to come up in potency or antigen level, but you're also bringing up a lot of other nasty things, including toxins. So I'd say the only way that to have a realistic chance of working is you'd have to go back and make all the seed strains all over again and knock out pneumolysine, and there's PSP. There's a couple other toxins that are there as well that you'd need to actually destroy. So you have to go back to the beginning to do that, perhaps 23 times over, right? So that's the difficulty in kind of a wholesale approach.
I'm sorry, just one more question. And why are you using the polysaccharides again?
Because they're known to be protective.
So they're an adjuvant?
They're not an adjuvant per se. In fact, they're very poorly immunogenic, but they are defined. It's well known that if you can achieve certain titers against those polysaccharides, that they correlate with protection beautifully. So if we know we can exceed a certain titer by an ELISA or an OPA assay for looking at pneumo, we know that that's associated with protection. To your point, there's other companies that are trying non-polysaccharide approaches. Novartis had a program. They had, I believe, 4 protein antigens. They were going to go after a protein-based approach for pneumonia. They discontinued their program last year. Sanofi still, I think they have 4 antigens as well, protein antigens. I don't know where they are with their protein. There's a local biotech company called Genosha that has, again, I think it's 4 protein antigens. It's a very tough road to follow because although these antigens are present in the nose where they stick and colonize, when they go systemic, these bugs do exactly what they do. They cover themselves in capsid. And if you look at fresh preparations of strep pneumonia coming out of someone's blood who's bacteremic, you can see the sugar right on the agar petri dish, right? Now, if you passage those things a long time in vitro, that sugar slowly goes away and the protein antigens sort of show themselves. But I think it's a tough task to actually follow that approach. And I'll tell you, more importantly, just as I mentioned, there are titers you can get from strep pneumonia against the capsules that correlate with protection. To do an efficacy study with a protein-based vaccine, you have to do a non-inferiority study. So if you look, we're at Prevnar 13. Pretty soon Merck's going to have a 15-valent. So wherever you go to do your efficacy study, you're going to have to find a place and have to include whatever is the state-of-the-art commercial vaccine and show that your titers are actually, that your protection level is at or exceeds that level. That's a gigantic study to do. I personally think my bias is the best vaccines out there, and could the Phoenix people please leave the room for a moment? No, would be polysaccharides from strep pneumonia and protective antigens, protein antigens from strep pneumonia, right? Everyone's always done heterologous crimp. Crimp, don't get me wrong, it works beautifully because it has T help. But a protein antigen that also provides some protection, I would argue, at least from an efficacy point of view, it might be better. Now, it invokes a whole other set of questions about safety, right? Have you detoxified pneumolysine well enough? We all know that CRIM is a tremendously safe molecule. So you're invoking risk on the safety side again. But again, if you're from a pure efficacy standpoint, I think a combination of the two approaches would be best. It's my personal thinking, right? Yeah?
Does the polycation that you have, I understand you said you have it there to just increase encapsulation efficiency. Does it affect immunogenicity?
Oh yeah, that's one of the bullet points that I showed.
Oh, I'm sorry.
Yeah, that was very much to our surprise when we hypothesized that it would help for entrapment. It did, and we get better particles. We can even have lower amount of reactants in the reaction. It all works as expected. What was a pleasant, unexpected result was when we look at a given amount of polysaccharide in a PCMV, with or without, again, compensating for what's been entrapped, they were substantially more immunogenic with the polycation that was actually used, and in fact still present in the matrix.